Assalamu alaikum. So this is the last lecture of the enzyme series. And in this lecture, we'll talk about cofactors. So the idea is that enzymes can carry out reactions using different catalytic strategies. We talked about some of these strategies like the acid-base reactions, the covalent catalysis, and so on. Now, some enzymes, many enzymes, rely on the amino acids that they have in the active sites. Okay, but and some of these enzymes they are almost polar. Almost all of them are polar amino acids that participate in enzymatic catalysis. Okay, the reason is that they are reactive. Okay, whereas non-polar hydrophobic amino acids are silent. They are stable. They do not catalyze reactions. They do not carry protons or electrons to carry out reactions like nucleophilic attacks, something like that, and electrophilic attacks. Now, some of these polar amino acids include serine, cysteine, lysine, histidine. In some cases, you can have arginine, you can have lysine, you can have glutamate, aspartate, and so on. Now, many enzymes prefer histidine, by the way. The reason is that its pKa is near neutral, near physiological pH, and it can carry out acid-base catalysis easily. Now, other enzymes need cofactors, and cofactors include, or they are in general, non-protein molecules. Okay, and when a non-protein molecule becomes part of a protein, we call this protein a holoprotein. In case of enzymes, now we call them holoenzymes. Without this cofactor, without this non-protein group, we call it an apoprotein or an apoenzyme. Now, what are these cofactors? Now, these cofactors can be divided or classified into three types. You can have metals, you can have small organic molecules, and you can have protein-based molecules. We're not going to talk about protein-based molecules in these lectures. We'll focus on these two, particularly the small organic molecules, which are also known as coenzymes. Okay, now the thing is, when these coenzymes bind tightly to the enzyme or to proteins in general, we call them prosthetic groups. Okay, just like binding of heme to hemoglobin, that makes heme a prosthetic group. Now, if the coenzyme binds loosely to the enzyme, then we call it a co-substrate. Okay. Now, in case of metals can bind tightly, and in this case, we call the protein. Okay, not the cofactor, the protein, we call it a metalloprotein. If the metal binds loosely, then we call the protein a metal-associated protein. Okay, so here we're talking about the naming of the cofactor itself. Here we're calling the protein. Okay, but anyhow, when a protein binds to a cofactor, anyhow, we call it a holoprotein. Now let's focus on these coenzymes right here. Now these coenzymes can catalyze two types of reactions. They can catalyze activation transfer reactions, or they can help enzymes in catalyzing oxidation reduction reactions. So now this is a table that shows the coenzymes or cofactors that we will talk about. These are mainly water-soluble vitamins. Okay, and notice that we're talking about vitamin B1, thiamine, vitamin B2, riboflavin, B3, nicotinic acid, vitamin B5, pantothenic acid, vitamin B6, pyridoxine. We're talking about biotin, folate, vitamin B12, lipoic acid, which is basically a fatty acid, ascorbic acid, or vitamin C. So when we finish this lecture, you can go back and refer to this table to summarize the naming of these vitamins and the type of chemical reactions that they catalyze. So in terms of activation transfer coenzymes, now the thing about these enzymes is that the functional group of the coenzyme directly participates in enzymatic catalysis. Okay, so they're not only facilitators, rather they do catalyze the reaction. Now the thing is that the coenzyme by itself is really inactive. It cannot catalyze any reaction. It's rather the enzyme, the protein, the amino acids that surround the coenzyme that enable coenzymes to catalyze these reactions. So the enzyme itself provides specificity as well as the catalytic power to the coenzyme. Okay, so these coenzymes depend on enzymes for specificity and catalysis. Now there are two other characteristics as well, and that is there are two groups in these coenzymes, two important groups. Now one of these groups is really the functional group. This is the one that catalyzes the reaction and it forms a covalent bond with the substrate. Now the other group is the binding group. This is the group that connects the vitamin or the coenzyme to the enzyme itself. Okay? So let's talk about the first one, vitamin B1 or thiamine. Now thiamine by itself is not really active. It has to be activated, and it's activated by phosphorylation. That is, you have the addition of pyrophosphate. That is two phosphates. Okay, so this is thiamine right here, and by adding a pyrophosphate, that is these two phosphates, then it becomes an active coenzyme, and it's known as thiamine pyrophosphate or TPP. Now this vitamin is important in brain and liver, and this coenzyme is important for decarboxylation reactions, that is removal of CO2 from substrates. Now, if you look at the structure of the thiamine pyrophosphate, there are two groups that are important. The first group is this thiamine carbon right here. It's this carbon between the sulfur and the nitrogen. Now, this is the functional group. This is the one that catalyzes the reaction. Now we have another group that binds to the enzyme, the binding group, and the binding group is actually the pyrophosphate. Now what the pyrophosphate does is that it's a chelating group. What we mean by this is that whenever you have the formation of when you have a group that forms two covalent bonds with a metal like magnesium, this is known as chelation. Okay, now this magnesium is now the group that is linked to the enzyme. So you have the pyrophosphate now chelated or chelating magnesium ion, and the magnesium connects the thiamine pyrophosphate to the enzyme itself. So that's the binding group, right? Now this is the functional group. So what happens here is that the thiamine carbon, what it does is that it binds to the keto group of substrates, releasing CO2. So right here you have the thiamine carbon right here, and this is pyruvate. This is one of the most famous reactions that convert pyruvate to acetaldehyde. Now what happens here is that you have this carbon right here that forms a bond with the carbon, the keto carbon, right? And you have electron rearrangement, and that results in the release of this carboxyl group right here, carboxyl group of pyruvate, in the form of CO2. Now we have a 2-carbon molecule right here. You have other electron rearrangement, and that results in the release of this 2-carbon molecule, or the acetyl group, okay, or acetaldehyde. Now, so thiamine pyrophosphate is important for decarboxylating pyruvate. Okay, so you have this reaction that we previously talked about. It's the reaction catalyzed by pyruvate dehydrogenase complex, if you remember. So this is a complex that catalyzes actually 3 reactions. One of them requires TPP. It's the very first reaction. And this TPP, again, it results in the release of CO2 from pyruvate, okay, resulting in the formation of the acetyl group right here. Now, notice something here. This pyruvate dehydrogenase requires TPP, and it also requires other cofactors that we will talk about in this lecture. Okay, so pay attention to that. So we're talking about FAD, NAD, lipoic acid as well, okay, and TPP, and as well, you know, the coenzyme A, so vitamin B5. So pay attention to that, okay? So that's one reaction. Another reaction is the alpha-ketoglutarate dehydrogenase. Same thing, you have decarboxylation of the alpha-ketoglutarate. So this is the ketone group that binds to to the thiamine carbon, and this CO2 is the one that is released, resulting in the formation of succinyl coenzyme A. This is an important reaction in the Krebs cycle. Now, again, the mechanism of action is that this thiamine carbon forms a bond with the keto carbon right here. This is followed by electron rearrangement and release of CO2. Okay? Now, let's talk about vitamin B5, or which forms coenzyme A. Now, vitamin B5 is known as pantothenic acid or pantothenate, and this pantothenic acid is really formed from 2 groups: pantoic acid, okay, which is this part of the molecule, as well as beta-alanine. Beta-alanine. Why? Because why is it called beta? Because usually you have the nitrogen coming out of this carbon right here, the alpha carbon. So but you have now this nitrogen extending from the beta carbon. That's why it's called beta-alanine. Now pantothenic acid is important for the formation of coenzyme A, and coenzyme A is this large molecule. Okay. So what you have in here is an ATP that is added to the molecule. Okay. Now, part of the pantothenic acid is modified, and you have the addition of a sulfhydryl group to the molecule. Now, so you have again 2 groups. You have a binding group that binds to the enzyme, and you have the functional group, the catalytic group. And the binding group is actually the ATP. Okay, and the catalytic group, or the functional group, is the sulfhydryl group. So this part binds to the enzyme and gives specificity to the molecule as well as to the enzyme. And this is the small group that catalyzes the reaction. Now, coenzyme A is important in the metabolism of carbohydrates, fats, and proteins. Now, the enzymes that require coenzyme A is, again, pyruvate dehydrogenase complex. Okay, so this reaction involves pyruvate reacting with coenzyme A, now coenzyme and NAD. Now coenzyme A you have a decarboxylation reaction forming an acetyl group, and this acetyl group is attacked by the sulfhydryl group of coenzyme A, forming acetyl coenzyme A, and you have CO2 as well. Another reaction is the citrate synthase reaction. In this reaction, you have conjugation of oxaloacetate with acetyl coenzyme A. This is the very first step in the Krebs cycle. Okay, we have the formation of citrate. That's why the Krebs cycle is also known as citric acid cycle. Okay, so this reaction right here requires vitamin B5 as well. So you have the coenzyme A as well, attacking this coenzyme A here, attacking the oxaloacetate, resulting in the release of the coenzyme A and conjugation of the acetyl molecule with the oxaloacetate, forming again citrate. Now, the other enzyme that catalyzes these activation transfer reactions is vitamin B6, also known as pyridoxal phosphate. So pyridoxal phosphate is the active cofactor, which can be derived from these precursors: pyridoxine, pyridoxal, pyridoxamine, depending on the functional group that exists on the ring. Now, this vitamin B6 is important for transamination reactions, and these reactions are reversible. We already talked about these reactions. So these reactions involve transfer of amino groups from amino acids to keto acids, resulting in conversion of amino acids to keto acids and keto acids to amino acids. So what you have in here is this transamination reaction catalyzed by alanine transaminase. You have a transfer of amino group from alanine to alpha ketoglutarate, the keto acid. Now the keto acid is converted to glutamate, and alanine, when you remove the amino group, is converted to pyruvate. And you have aspartate transaminase as well. This enzyme also requires vitamin B6. Again, transfer of amino group from aspartate to alpha ketoglutarate. Alpha ketoglutarate becomes glutamate, just like in this reaction, and aspartate becomes oxaloacetate. Okay, so here you can link, by the way, sugar metabolism to amino acid metabolism. Okay, so again, you will learn all these reactions next semester, inshallah. So here you have the ring structure, the aldehyde group. It constitutes both the binding group and the functional group as well. So what happens is that this aldehyde group forms a covalent bond with the amino acid, extracting the amino group from it by electron rearrangement, including groups within the ring itself, and release once it's taken. The keto acid is released, and then you have entry of the keto acid, and you have the other keto acid, and the other substrate. The first substrate is the amino acid, okay, which gets converted into keto acid. Then the keto acid comes in. It binds to the amino group, electron rearrangement, so you have the reverse of these reactions and release of the amino acid. Now, the other vitamin is vitamin B7, or which is also known as biotin. Now, this is the structure of biotin, and biotin is involved in carboxylation reactions, that is, transfer of carbon to molecules, making them larger by one carbon. Now, this biotin, when the active form of biotin, or biotin, becomes active when it binds to lysine within the enzyme. Now, the thing with biotin is that its main source is food as well as intestinal bacteria, so we cannot synthesize biotin. And we hardly have deficiency of biotin because intestinal bacteria can synthesize it for us. And that is why people hardly suffer from biotin deficiency, but they do when they take a lot of antibiotics for a long time, or when they eat a lot of raw eggs, because raw eggs contain a protein known as avidin, and avidin binds to biotin. Now, enzymes that need biotin include pyruvate carboxylase, catalyzing this reaction. Pyruvate it becomes larger by 1 carbon, so you have transfer of CO2 to pyruvate, converting into oxaloacetate. Another reaction is acetyl coenzyme A carboxylase, which is a reaction that is part of the fatty acid synthesis metabolism. Now, let's talk about the oxidation reduction coenzymes. Now, in this case, now these coenzymes work with oxidoreductases, and they help oxidoreductases catalyze these reactions. Now, these reactions, these coenzymes have their own functional groups. Okay, so, and these functional groups can accept and donate electrons. And these electrons can be in the form of hydride ions, hydrogen atoms, or oxygen. Now, one huge difference between oxidation reduction coenzymes and activation transfer coenzymes is that oxidation reduction coenzymes do not form covalent bonds with the substrates. Okay? But they can bind to the enzyme though. Now, the most common coenzymes include NAD, which is derived from niacin or vitamin B3, as well as FAD or riboflavin, which is derived from vitamin B2. Now, there are other vitamins that can also catalyze oxidation reduction reactions, like vitamins E and C. And in this lecture, we'll talk about vitamin C as well. Now, again, these coenzymes need enzymes for additional specificity and catalytic power. So let's talk about FAD and FMN. Now, these two are derived from riboflavin. Riboflavin is also known as vitamin B2. It can be modified by the addition of a phosphate group, and it becomes it's known as FMN, or flavin mononucleotide. Now, if you have the addition of ATP to this phosphate, then you have what is known as flavin adenine dinucleotide. Okay, so that's the difference between FAD and FMN. And this is really the reactive group right here. This is the one. Okay, and what happens here now? So enzymes that contain riboflavin or FAD or FMN, they are also known as flavoproteins. Now, one thing important about FAD is that it accepts electrons in the form of hydrogen atoms, and these are donated sequentially, meaning that you have one electron or hydrogen is bonded to this nitrogen, and the second one is bonded to this nitrogen right here. So the two hydrogens, by the way, can come from one source or two different sources. The thing about FAD and FMN is that they are involved in reactions that result in the formation of double bonds or disulfide bonds. So keep that in mind when when you go into the metabolism course. Now, these coenzymes FAD and FMN are prosthetic groups. So what are prosthetic groups? Prosthetic groups are coenzymes that bind tightly to enzymes, okay, or to proteins. Now, why do they bind tightly? It's a protective mechanism. Why? Remember how I said that these electrons are transferred sequentially? So what happens here is that when you have this group right here accepting the first electron, that converts the coenzyme into what is known as semiquinone. The semiquinone is a radical, and radicals are dangerous. They are risky, they are harmful. They attack other molecules and they damage them. Okay, so in red blood cells, they can damage hemoglobin molecules, damaging them. They can damage plasma membranes, killing cells, and so on. So these radical molecules are dangerous. So by having FAD and FMN as prosthetic groups, they are tightly bound. They are enclosed. They are imprisoned. They cannot attack other molecules, and they cannot attack other molecules, and you have the other electron now accepted by the second nitrogen group right here. So that's why they are prosthetic groups. Now, examples of reactions that require FAD, you have succinate dehydrogenase. This is a reaction in the Krebs cycle. Note here that you have the formation of a double bond when succinate is converted to fumarate. So let's talk about NAD and NADP. Now these two are precursors of vitamin B3, and they and this is the structure of niacin, and that forms nicotinamide adenine dinucleotide. When you have the addition of a phosphate, now it's known as NADP. Okay. Now both of these are co-substrates. Okay, so they are not tightly bound to enzymes, rather they are loosely bound. And they are important for dehydrogenase catalyzed reactions. So here you have the structure of the NAD. That's how it looks like. Now the thing is, the difference between NAD and NADP is the addition of a phosphate group to this portion of the molecule. Now this portion is not really important for the catalytic part of the coenzyme at all, but it's important for the interaction between NAD and NADP with the enzyme. So the phosphate determines specificity of binding rather than the catalytic action itself. Now the activity of NAD is determined by this group right here, by the niacin ring itself. This is the group that can accept or donate the protons, or electrons I should say. Now NAD and NADP are involved in oxidation of alcohols and aldehydes. Okay. Again, remember that for the metabolism course. So one enzyme that requires the function or the presence of NAD is lactate dehydrogenase. We talked about lactate dehydrogenase a lot previously, and this enzyme catalyzes the oxidation of lactate to pyruvic, and that accompanies reduction of NAD to NADH. Now the same enzyme can reduce pyruvic to lactate, and NADH is oxidized to NAD. Now, in the active site of lactate dehydrogenase, you have histidine. So this one right here is histidine. This is lactate. And you have an arginine that is part of the active site, and here you have the niacin part of NAD. Okay, so what happens here is that this histidine can attract electrons towards it, and at the same time that makes it easier for NAD to extract electrons in the form of hydride ion into this functional group right here. So you have transfer of electrons, and you have the formation or conversion of the alcohol group, the OH, into a ketone. So now it gets oxidized. Now vitamin C is also an enzyme that involves in oxidation reduction reactions. And vitamin C is also known as ascorbic acid. It's important for the function of prolyl hydroxylase, which is important in the synthesis of 4-hydroxyproline, which is part of collagen, right? And it is also an antioxidant. So the reaction involves, again, hydroxylation of proline to, right here you have the formation of hydroxyproline, okay? Now, the thing with ascorbate is that it is an antioxidant as well. So what it does is that it can extract electrons from harmful molecules, from radicals. So the ascorbate itself becomes a radical by itself, okay, when it extracts electrons, when it gets oxidized into dehydroascorbate. Now, the oxidized form of ascorbate is stable. It's not reactive. It doesn't cause cellular damage, and it helps our systems and cells from getting rid of radicals and reactive oxygen species. Now, the thing about this molecule that makes it a favorable antioxidant is that it forms a resonance. What we mean by that is that you can have this electron extracted from the radical molecule into the ring structure, and it keeps rotating, it keeps running in circles. Okay, and that is really important. This is resonance. Okay, this is really important because if it runs in a chain, at the end of the chain, it has to jump to another molecule, damaging it. So now you have the electron trapped, running in circles inside the ring. So let's talk about metals now. Now, we said that metals can be part of enzymes, and they can catalyze different reactions as well. Now, these metals include magnesium. We talked about that. Okay, and magnesium, by the way, is important for the function of hexokinase. You also have zinc. Zinc is also important for the function of other enzymes like carbonic anhydrase, and we'll talk about carbonic anhydrase in a second. Now, these metals, they can act as electrophiles. Okay, so they can attack other molecules, extracting electrons from them. They like electrons. Now, they can also help in binding to substrates, and they can stabilize anionic structures during the reactions. They can also accept and donate electrons in oxidation reduction reactions. So they're really important for multiple enzymatic reactions. Now, metals have advantages, okay? One of them is that they carry positive charges, okay? And that makes them able to form strong bonds. But at the same time, these bonds, even though they're strong, they are labile, meaning that they can be kinetically changed. The other thing is that they are stable in more than one oxidation state. So iron, for example, it can be in the ferric form or the ferrous form. They can bind multiple ligands in different coordination spheres. It can form multiple covalent bonds. So magnesium, for example, we talked about that before. It can form, it can be chelated into phosphate groups, right? The phosphate groups of ATP can also form these links with magnesiums as well. So let's talk about carbonic anhydrase. Carbonic anhydrase is an enzyme that converts CO2 and water into carbonic acid. So what it does is that it hydrates CO2. Now, this reaction, by the way, it can occur spontaneously. Okay, that is without the need of enzymes. But what carbonic anhydrase does is that it accelerates the reaction further. And its importance stems from the fact that mutations or deficiencies of carbonic anhydrase can cause osteoporosis as well as mental retardation in individuals. So that indicates that carbonic anhydrase is an important enzyme. So zinc is found in the +2 oxidation state in biological systems, and in carbonic anhydrase it can form covalent bonds with imidazole rings of 3 histidine residues. Okay, now it can also form a 4th bond with a water molecule. Now this is what zinc does to the reaction. So it forms these covalent bonds with 3 histidines. It can form a 4th one with a water molecule. That results in the release of 1 proton, resulting in the formation of a hydroxide ion. Now this hydroxide ion is now very reactive. When CO2 comes in, the hydroxide ion can react with the carbon of CO2, resulting in the formation of a covalent bond between the two, and then you have electron rearrangement, and that results in eventually the release of H2CO3 from the molecule, and then of course it gets ionized quickly into bicarbonate ion. Now, zinc is part of the active site of alcohol dehydrogenase. Okay. Now, alcohol dehydrogenase catalyzes the conversion of alcohol into acetaldehyde. So you have alcohol being oxidized into acetaldehyde, and at the same time you have NAD being reduced into NADH. Okay. Now, zinc is part of the active site of alcohol dehydrogenase. Now, contrary to carbonic anhydrase, zinc does not participate in the catalytic reaction itself. So it doesn't form a covalent bond with the substrate. Rather, what it does is that it facilitates the reaction. It makes it easier to happen. So in the active site, you have alcohol now N. And this alcohol can form these non-covalent interactions with serine within the active site. Now, near serine, you have histidine as well. Now, what histidine does is that it pulls protons from the serine. Now, serine can pull protons of the substrate's OH group. So it pulls this proton towards it, okay, to compensate for the pulling of these electrons away from it by histidine. Now, that results in the formation of oxygen with a negative charge. In order to stabilize this oxygen, now you have the formation of this interaction with zinc. Now this oxygen is stabilized. At the same time, you have NAD coming into the active site, and it can extract hydride ion from alcohol molecule into the niacin ring, right? So now NAD extracts protons or electrons from the alcohol molecule, and it becomes NADH. So what zinc does is that it stabilizes the molecule, the transition state, the alcohol molecule, and the interaction between the substrate with the active site of the enzyme.